sure it's facing off. Maybe a little higher. And our last talk of the day is entitled Cardiac Effects of E-Cigarettes by Jorge Espinoza Derout.
Thank you for coming. The title of my presentation is Electronic Cigarette Expose Induced Cardiac Dysfunction in apoino mice. Well, our laboratory is interested in the how electronic cigarettes affect the cardiovascular system. Electronic cigarettes are growing very importantly in the population that is in college and high school students. Although there is not much information about which are the effects of electronic cigarettes in the cardiovascular system. Many regulatory agencies assume that electronic cigarettes are safe, and five of them contain nicotine that is well documented as it has effect on the cardiovascular system. So what are our specific aim for this work? The specific aim is mainly to determine whether the e-cigarette vapor promotes cardiac function and after crosses in the apoino cow mouse model. In this work, we use the channel that Dr. Matt Chow already designed. And why we choose the apoino model? The apoino model is a major model that is used for studying atherosclerosis. So the first question we did is basically we put the mice 12 weeks in the chamber with that we smoke 12 hours in cigarettes. One of the most common measures to analyze the function and the structure of the heart is the echocardiogram data. So what is important here? Well, basically you can measure morphology and measure function of the heart. So when we see markers has the left ventricular mass, we need to observe major changes in the morphology of the heart. However, when they observe markers like to a specific measure like to fractional shortening, as measure of contractility of the heart, or ejection fraction, or the measure of contractility of the heart, we see a decrease in ejection fraction and a decrease in fractional shortening of the heart. Therefore, the mice heart is contracting less after 12 weeks of exposition with the electronic cigarette. So basically, what is the theory was the electronic cigarette induces a dysfunction in the contractility of the heart in the mice. So here we go with a technique a little bit more complex. Basically, we do something like a RNA-seq analysis. We take basically frequent everything of RNA in the sample. We take a small piece of the ventricle and we sequence 225,000 genes and we compare the controls mice with the electronic cigarette mice. And from these genes, we observe one of the genes that change. In this graphic, I show the two-dimensional helicopter of these 113 genes. The one who are in red and the one who are in green decrease. And when we go to analyze these genes, specific genes, with the different chemical real-time PCR, we observe that the genes that are increased, some of them are like to collagen 5, C4, and TNF receptor superfamily, and these genes are markers for inflammation. As well, we see a gene like to harakiri, but it's a apoptotic gene, and as well other genes that we observe, family of genes related with the circadian rhythm of metabolic changes, like two very well-known circadian rhythm of metabolic changes genes are PAR2 and PAR3 and MPAS2. Coincides as well with certain genes, like to ST1, STB1, that they are genes related to the increase in the production of fatty acids. So when we get all this data, we put in a program that analyzes which are more representative pathways that are changes. We use this program called engineering pathway analysis to analyze all the data and then which one are the pathways that are really deregulated. And the pathways deregulated mainly atherosclerosis signaling, this rim signaling that are related with metabolism, and leukocyte translation signaling. Therefore, this data, we have RNA data that tell us that electronic cigarette induce inflammatory phenotype, and this phenotype match the functional data derivative from the echocardiographic data. So now, one of the main diseases that people know that related to inflammation is atherosclerosis. When after the heart is taken from the mouse and the aorta is staining with the one of the staining, because it's a staining for lipid deposition. So we see that these are the cigarette mice and these are the contours. And these are atherosclerotic plaques of the mice. And we observe an increase in the atherosclerotic plaques in the mice that were exposed to electronic cigarettes. And here's the quantification of data that's a clear increase after 12 days of atherosclerotic plaques in the ApoE mice exposed to electronic cigarettes. Therefore, we can see from the data that the electronic cigarette had a strong atherosclerotic produce increased atherosclerosis in this model. So what's the conclusion of data? ApoE mice exposed to e-cigarettes have a decreased fractional shortening and ejection fraction in comparison with control ApoE mice. This result implies a novel effect of e-cigarettes regarding the function of the mice contract their heart. Besides, RNA-seq analysis showed that the deregulation of pathway related with atherosclerosis, circadian rhythm, leukocyte translation signaling, and this is together with this point of phenotype. And as well, we know that ApoE mice exposed to electronic cigarettes have an increased atherosclerosis lesion progression in comparison with the ApoE control mice. Now, currently, we are performing the experiment to study more on the molecular level what are the changes that induce this phenotype, this inflammatory phenotype. So I would like to thank the people who work with us, my advisor, Dr. Friedman, and Dr. Friedman Laboratory, Dr. Sinha Hicking Laboratory, Dr. Max Shaw, that developed the machine, and the grant that we support our work, and for the mind who they have in this job. Thank you.
Any questions for Jorge? I got a question sort of from Max and Jorge together. Based on your delivery model between the vapor and the aerosol, do you think that it's possible that e-cigarettes could be worse than conventional cigarettes in terms of getting the concentrations into the lungs, into the circulation?
We don't know yet.
Right.
So actually, that's important to then in the future compare normal cigarettes in the chamber with the e-cigarette and see the same changes that we are checking now to see what is different. So this will be a project.
Great. Well, thank everybody for staying. So we do have an award for the best training presentation, and we're going to give it to Zach Harney. On full method of energy self-administration. Again, how's that? Exactly. You do the right answer. And you guys have any comments for next year, something afterwards. Keep it as really enjoy today, and then you're ready next year. Thank you. Evaluation, back there. If anyone has one you haven't done, you know, then that's off. Good question, though. Thank you very much. Full day. Ready to hire? Very well. Thank you so much, Dennis.
You're quite welcome. You're all disconnected.
I think it went well.
I think so too.
Yeah.
You like the new system?
Yeah.
I think it worked really well. The nice part is it's not hard to set up.
No.
You don't have big speakers in the way, and
Maybe this could have been a little bit bigger there, but maybe this preview could have been a little bit bigger, but it was okay.
yeah. Let's see.